John Nicholson from Wellcome Trust Sanger Institute on iRODS at the Sanger Institute.
Cool. You've got your own laptop. All right, I don't know on there.
You can get it close enough.
Looks all right. Fine.
It's going to record your computer.
Okay, so okay, record that.
I think it's recording this. Yeah, so there's a way to do.
I'll have to change that. Going to do the right thing.
Sorry about that.
Oh no, that's done the wrong thing. Okay, change the way it works.
Yeah, as I understand it, the recording is picking up what's on your computer. So if you've got yours in presentation mode and you're going to give a talk, they're going to see what's on your screen, not what's being projected. So don't do it that way.
Right, that should be the same now.
Okay.
Hi, good afternoon, everyone. I'm John Nicholson. I work at the Sanger Institute in what's known as the Informatics Systems Group, although we're trying to rebrand ourselves as the High Performance Computing Group. I've actually been at Sanger quite a long time. I've been there 19 years now, although I've only been in my present role for the last year. And I've only been playing with iRODS, really, for the last sort of 3 or 4 weeks when I knew I was coming to this. But before that, I have actually been involved in sort of setting up the hardware and the networks that are used by all the Institute running the iRODS systems. So today I'm going to give a bit of an overview of the Sanger Institute and how our data flow works, and then how we use iRODS within that infrastructure to manage our large amounts of data, a little bit about how we're safeguarding all the data that we've got at the Institute, and how we're going to expand that in the near future. And then if we've got some time, I'm going to talk about some of the other projects we're involved in that are going forward into the future. So the Sanger Institute was set up in 1993 to be the UK's arm of the Human Genome Project. And we actually were the largest single contributor to that project, contributing about 1/3 of the total data to the project. Obviously, since that was completed in about 2003, we haven't been sitting on our laurels. We've been taking part in lots of different projects around other areas. So we have a project around what's referred to as model organisms. So these are mice and zebrafish sequencing are the most common ones. We have a big cancer genomics project, as well as ongoing human genetics and variation. We have a large program involving for pathogens and cellular genetics. And alongside this, we also have a large bioinformatics team who are developing codes and analysis programs that are used not only at the Institute, but all the other places around the world. So things like SAM tools are actively developed at Sanger. Underpinning those core projects, we have some core facilities. We have a DNA pipelines group whose responsibility is to basically do an industrial type sequencing so that scientists just supply their samples and get the raw sequence out the other end without having to worry about any of the running the sequences, running the sequencing laboratory. Our model organism program, we have a breeding program where we supply mice and zebrafish for use at the Institute and other research organizations. We have a cellular generation and phenotyping laboratory where we generate iPS cells, induced pluripotent stem cells, from samples donated by patients or volunteers at the local hospital. And of course, underpinning all of these is the IT infrastructure, including our data center on campus, which is about 1,000 square meters, hosting about 20,000 cores of compute and about 26 petabytes now of storage. So previously, my colleague Pete Clapham presented at the iRODS group in 2013 at Supercomputing. And so I'm going to recap a little bit of what he talked about here and go on from there. He also made some predictions about where we'd be going, and we'll be filling in on some of the whether those have come to fruition for anybody who was at that meeting. Within the next generation sequencing pipeline that we run, so within our sequencing pipeline, this is how the data flow actually sort of works. The data comes in from the sequencer and lands on a staging server in the data center. There, the initial analysis is run, so we do some quality control, and the initial alignments are run, and then that data is put into our iRODS system. That's sort of the first pass of analysis, and that's where the sort of sequencing pipeline stops, and then it is made available through iRODS for further analysis at a later date. When the second stage analysis wants to be carried out, data is taken out of the iRODS system and staged onto our Lustre file system. Lustre, for those of you who don't know, is a high-performance parallel file system, which we run across a 10 gig Ethernet network within the data center. We're just upgrading that to 40 gig, actually. And then the second stage analysis using another compute farm with about 6,000 cores runs on that, and whatever codes the scientists need to run on that are run. Once that data is completed, it's either shoved back into our iRODS system for archive, again talking about preserving this data for future use, as was just being talked about. It's also submitted to external databases, things like the EGA database hosted by the EBI, or it's provided directly for consumption on our website through things like the Ensemble browser. The staging servers that collect the data from the sequencing machine, this is our sort of first generation of these staging servers. They're basically 2U boxes, a bit like the HP DL385s or equivalent from Dell or whoever, with about 50 terabytes of direct attached storage via fiber channel. There's one of these per sequencer, and they just present that 50 terabytes of storage out as a Samba share so that the Windows box that's attached to the sequencing machine can write directly down to this disk. It's then exported via NFS, again, to a 1,000-node compute farm for this first stage analysis. And that just reads and writes over NFS. It does all the QC and referencing to the alignment to the reference. And then when it's finished with that, the BAM or CRAM files are stored into iRODS. We're actually looking to replace those nodes with a sort of slightly simpler model, because we can now get these machines. These are based on the HP SL4540 machines. These are machines that can take a large number of disks, so we can actually put 50 terabytes or more disk space into the machine and use a SAS attachment, so we eliminate the need for any sort of complicated fiber channel infrastructure. We can get quite a decent chunk of CPU and memory into these boxes, so we can eliminate now the 1,000-core compute farm and run the first stage analysis on these nodes directly, eliminating the need for NFS and also making our sort of failure domain a lot smaller. So if we have to take the 1,000-node compute farm down for some reason, software upgrades or whatever, we basically have to stop our sequencing pipeline. With these, if we have to do an upgrade on one of these machines, we only affect one sequencing machine, and we can probably tie that in with sort of routine maintenance in the sequencing facility. In terms of our iRODS, this is how we've laid everything out. We have one portal zone. This slide was actually sort of produced for people who may not know much about iRODS, so there's a few bits up there that I don't think I need to explain to this crowd. We have one portal zone, and then underneath that we have all the different zones for the different research projects that are going on. So we've got our sequencing zone, human genetic zone, and the UK 10K zone, as well as our archiving zone. Each zone has different rules based on the scientific need for that particular project. So in this example here, the UK 10K project have decided that they only actually ever need to have one copy of their data, so none of their data is replicated. All the other groups have said, we're prepared to pay for our data to be replicated, and so it's replicated between our red and our green data center suites. At the moment, we try and keep the rules really light, so the only rules that are actually there are to do with replication and checksumming. We use checksums all the way through the application. So the application will generate a checksum, and then as the data goes into the iRODS system, that checksum is checked to make sure that the data has been properly committed down to disk. Physically, this is what we've got. Each zone consists of 2 iRes servers. This is how Pete left it back in 2013. Active iRes servers running against large disk arrays from something like the DDN SFA 10K. Although we do have some other vendors' storage in there where we don't need the same level of performance, and we need just basically bigger storage at lower cost. The iRODS servers have 8 gig fiber channel down to the disks, and they present this out to the network with 10 gig Ethernet. We're using Oracle RAC for our iCAT server. We do that. We use Oracle rather than any of the sort of free databases because we already have a large Oracle install base on campus. We have a site license for Oracle, and we have a large DBA team with a lot of Oracle knowledge, so it makes a lot of sense to leverage that to make our databases resilient. So that's sort of considered our pass one iRODS servers. It's quite a large failure domain. We've got hundreds of terabytes underneath one iRODS server. If that goes down for any reason, we lose access to some of that data. It's not a terribly good idea. The bandwidth is starting to become a bit of an issue. We've got hundreds of clients now trying to talk to each one of these servers, and even though we've got 10 gig connectivity, that's starting to be a bit of a bottleneck. Building out large arrays means we have to buy data in very large chunks, and that can get quite expensive if there are smaller groups who don't have such a decent budget. And having these large POSIX file systems, if there's anything does happen to it, it does take a long time to check them and bring the service back up. And the other problem with it is that both copies are actually still on the same campus, under the same roof. They're in different data center suites, so they have different UPS and different cooling circuits, but they are still in the same building. And that's a little bit of a problem based on where we are. We're based in a small village just outside Cambridge, and we're not very far away from Duxford Airfield, up here in the top left-hand corner. And I don't know if anybody knows Duxford Airfield, but that's part of the Imperial War Museum, and they quite often fly Spitfires and other 70 or 80-year-old aircraft out of there. And 4 or 5 times a year, they have a big air show where they have old aircraft as well as modern jets doing aerobatics, and they tend to like to do those directly over our data center. Now, accidents unfortunately do happen, and aircraft have fallen out of the sky, and it's considered only a matter of time before this has a possible impact on campus. 2 of these events actually happened within a month of each other, and that was actually sort of while the data center was being built. So it did give us a bit of a pause. So we decided that we should probably move at least one copy of our data off-site. So we could just we've already got 2 copies. We should be able to pick one of those copies up, put it on a truck, and take it to a new data center. Fine idea. But of course, that means we've actually got to go out and buy a second data center. A little bit of serendipity here. We were actually being involved in the Janet Shared Data Center project. This was a project between Sanger Institute, the Crick Institute, and several of the London universities, King's, Queen Mary's, UCL, to name a few, to basically come in together and procure a data center for them to move into. We all needed some more data center space, so it made sense for us to go together. We employed the Janet organization, that's the UK Academic and Research Network, to sort of broker that deal for us. And this time last year, we were going through sort of the last stages of tendering, and we finally decided on this data center at Slough, just outside London, from Infinity, and we've decided to move our data down there. Unfortunately, if we took one copy of our data off and moved it down to the data center in Slough, we'd be left with only one copy of our data available for the duration of the move. And with the best will in the world, it's going to take a couple of weeks to actually move that amount of data. We've got to decommission the servers, we've got to rack them up, put them on a truck, ship them down there, recommission them, and I'm running out of time. So that's what we did is, in the end, is we decided we'd build a 3rd copy of our data, and also at the same time trying to address some of the issues around the iRODS servers. So we've decided to use the same technology that we're using for this new generation of sequencing servers, and use these SL4540s as independent iRODS servers. We built the 3rd copy on site, we copied our data across, we shipped it all across to the Infinity Data Center, brought it all up, and now we've got these new servers down there. We're starting to free up one of the copies on campus to double our capacity on campus. These smaller nodes give us a sort of self-contained zone, so if we have any small projects that want to come along, we can just spin up a new one of these servers for them, and it's a lot cheaper than them having to buy a whole RAID array. With 30 of these machines, that's how many we deployed down at Infinity, we obviously get a lot more bandwidth, but there are some questions about whether this will be reliable. But so far, so good. We haven't had any problems with it yet. The copies all seem to go okay. We had a few issues at the actual installation, bits of bent metal that happened on the truck, but we've caught up and everything seems to be spinning okay down there now. So the future. It's always a bit difficult to predict, but these are some of the things we definitely know we're going to need. Better metrics and monitoring, security, including federated authentication as we start to get into more collaborative projects. Secure data. We're starting to get involved in medical projects, so where we're looking at patient data. So a lot of the topics that have already been touched on today. We're going to probably need to secure data in flight, so being able to send the iRODS data over SSL, and also at rest, potentially. So making sure that the data is encrypted on disk. And another thing that's always been touched on, we're going to need better access to the to metadata. So being able to do better searches against our metadata. I'm not really going to have enough time to go through some of these slides, so I'm going to have to skip forward. Another big project is Medlab. This is a collaboration project between ourselves and some of the universities in London and the EBI. This is to build a shared a shared space for people to run their bioinformatics codes against the large data sets that we're generating. And what Sanger are going to be contributing are basically access to our iRODS system. So being able to give all the data from our sequencing facility to our collaborators at the shared data center space, which itself is also going to be at the same data center down in Slough. At the moment, within Sanger, we're stuck on iRODS 3.3.1. We desperately want to get onto 4 because we definitely want some of the new features. However, it's a bit problematic for us because of all the changes under the hood. We have got a large amount of storage under iRODS now, 7 petabytes of storage under there. And so making any changes to that system does make us a little bit twitchy. We don't want to lose access to that for any sort of length of time. But that said, we are going to be doing it. We're going to be running a reevaluation project later this year to see how we can migrate onto 4.x later this year, because we desperately need to look at things like the PAM SSL authentication and get the hierarchical groups need to be sorted out because we make use of those quite a lot. We want to, as I say, we want to split out our metadata into an external database so we can use things like Elasticsearch. We want to also make use of the PAM module so we can use the Janet Ascent project, which is another federated authentication system. I've actually sort of sped up way too much now. So finally, I'd just like to thank a few people. Obviously, Paul Wooby is our Director of IT. He pays the bills. Tim and Pete, they put together these slides, so that's why I'm sorry. It's a bit of a quick run through because I didn't actually write these slides myself. And all the other members of the system, the ISG team. So James Beale and John Constable have been doing a lot on our storage side. Simon Fraser has been doing a lot with our federated authentication. Shanti is our Oracle DBA, and Mark Jarrett runs our infrastructure team. So basically make sure all the systems actually work. And obviously we have a load of bioinformaticians who write lots of code that break everything. So, thank you.
We have time for a question.
Yeah, sure. I will try and answer it.
I'll make it a simple one. Do you use iRODS to manage the movement of data between Lustre and your archival storage?
Good question. I would not be able to say that with 100% certainty. I'm pretty sure we do. It basically probably be just either using iPuts or actually direct API calls that we've written in-house. There's a suite of tools that we've written in-house called Batten, which basically extend a lot of the I commands, which we use internally.